So I'm going to talk a little bit about DNA information and the various applications on commercial cattle ranches. And I guess I should be a little perturbed that my other family members chosen not to be at this talk. That's not a good sign for those of you that thinking of what he might know that you guys don't. But my 13-year-old son must have gone on the nature trail. So I'm going to talk a little bit about the things I think are working well in DNA technology. We hear a lot of hype about DNA technology, and we have a lot of salespeople out there pushing DNA technology, and there are some spectacular success stories in terms of what it's been able to do for both the purebred and commercial cattle industry. And then there's some aspects that aren't working so well at the moment. It's a new technology. It's still getting its act together. It's a little bit, I guess, analogous to the early days of computers when we used to have the floppy drives, and I think we can all remember that looking back, they weren't particularly useful, but it was a technology that was in development, and now could we get by without all of the different technological things that computers have allowed us to do. And then I'll just touch a little bit on what the future holds. And I'm going to be talking a lot about a project that I've been working on with the commercial ranchers up in Northern California as kind of where my data is coming from. But the first thing I wanted to talk about particularly was the use of DNA information for the identification of single trait defects or recessive or single trait things like horns and coat color and genetic defects. Who in the room has ever done a genetic defect or a horns or a coat color test on their cattle? Not that many, okay. Yeah, I thought there'd be a couple. Well, I'm going to talk a little bit about genetic defects because there's something that have been in the news a lot recently, particularly with three very prominent defects that have been discovered in the Angus breed in the last couple of years. And we've had genetic defects in cattle for a long time, and really I want to emphasize the fact that it's not one particular breed that gets genetic defects. Every breed has genetic defects. What happens is if you use an animal a lot, such that there's a chance that you're going to have both dam and sire that have a common animal in their pedigree, then you have the possibility of a recessive, that is a gene that has to be present in 2 copies for it to be seen. You have the probability of a recessive defect being shown in the progeny of that offspring. So the more highly used animals are, the more likely it is that you're going to start to see genetic defects. And they've done some studies that show that all of us carry about 2,000 genetic defects, that's humans in the room, and we probably each are carrying 1 or 2 lethal genetic defects. And that's why there's such strong rules against intermarrying amongst your family, because those genetic defects are lethal. And so it's just really a matter of if you have very highly used animals is when you're going to be more likely to see these genetic defects come together. And I want to talk a little bit about, in the 20th century, the most prominent genetic defect, and quite an interesting story, was the Snorcher dwarfs. And some of you may have been around at the time, but there was a very strong drive to phenotypically bring animals down in size to be more efficient. And this was a classic example of a prize animal at that time. And this particular animal, whose nickname was Short Snorter, was seen as the type of bull that you would want to implement on your particular ranch. That was a bit of an unfortunate name because shortly after he came out they actually discovered Snorcher Dwarfism, which was a recessive genetic defect that was quite common in the Hereford and Angus breeds in the 50s, and it was actually traced back to an animal that was born in 1899, and so it took about 50 years for it to become evident that there was a problem with this particular genetic defect. And this is all really interestingly detailed in a book called The Battle of the Bull Runts that discusses about what happened here. But in 1956, a survey of Hereford breeders in the US found about 50,000 animals that had dwarf producing in the 47 states. So 50,000 animals that were affected that must have traced back to this one bull that was born in 1899. Now back then, the only way that you could deal with this was there was no genetic tests, and so the only way you could determine whether or not your animal was carrying a Snorcher dwarf recessive mutation was to do test crossing. If you test two carriers, I know every extension talk ever in the world does this example. If you test cross two carriers, what's the proportion of animals you'd expect to have the recessive defect? And that's 25%, right? And I know the students, the UCD students in the audience have gone through that a million times. And so you need to do that in order to be able to determine what the carrier was. And so there was entire lines of animals that were eliminated from the herd just because you couldn't be sure, and it was very expensive to do these test crosses. And it took a lot of effort on the part of the breed associations working with breeders and scientists to eliminate that particular defect from the herd. Let's compare that to this particular mutation, which is curly calf, arthrogryposis multiplex, which gives you this type of a phenotype. It's also a recessive mutation, and it basically, from a scientific standpoint, is a complete deletion of a segment of DNA. And in that segment of DNA is a protein that's required for muscle development. And when you eliminate that protein, then you get this curly calf appearance. And so one of these genes is expressed at a crucial time in the development of nerve and muscle tissue. Now this was traced back to a very prominent bull that was used in the Angus breed, and it was basically done by Dr. Stephan at the University of Nebraska in collaboration with Dr. Beaver at the University of Illinois, who in the period of about 3 months identified that there was this problem going on. They took some control calves and some affected calves like this, and as a result of having fully sequenced the genome of bovines, were able to identify the section that was missing in these calves, and were able to develop a genetic test in a 3-month period. Subsequent to that development, in the 10 months following the release of that test, about 90,000 cattle in the US were tested for this particular mutation. And the cost of the test is a little under $30, so obviously that's a lot of money. But have a think about what the consequences of that testing was. And if you look at what the results were, you've got 90,000 tests going on. You had certainly some carriers identified. There were bulls and there were heifers that were tested as carriers, about 18,000. But that left 72,000 animals that were free of the particular mutation that otherwise would have been suspect as a result of having the pedigree going back to the very highly used bull that was causing this particular mutation. And so those animals can continue to be in the herd and be used in the future. The carrier animals are not going to be a problem if you're crossing them to animals that don't carry the mutation. And so from a genetic perspective, you can still use those animals so long as you're aware of the fact that you don't want to be crossing a carrier with a carrier, but a carrier with a different breed or a carrier with a non-carrier will not give you affected progeny. And so there's ways to manage around that now that you know that your animal's a carrier. And so really the value that provided, if you I figured out the testing costs in the US, probably about $18 million in testing costs, but the savings in terms of animals that are free that could have been suspect is in the vicinity of about $200 million. And so it's a really huge return on investment when you look at those animals that are exempt from being suspect as a result of DNA testing. I kind of like this slide because this is the extension efforts that went on during the dwarfs in the 1950s, and there's this old slide here, and a phenotypic dwarf, and then we've got the carrier getting crossed with the carrier. The same old extension talk that we gave when we found the curly calf, and I think we have sometimes quite the same problems. And so I think some of you may have been in my talks where I do the clicker inputs, where the audience can click in as to their responses on this particular question. And so this is an example that I give quite frequently, where we're breeding a curly calf carrier cow, so an animal that's carrying that particular recessive mutation, to a curly calf free bull, so an animal that tested free, and then I ask what's the chance that the offspring will be stillborn as a result of being a curly calf. And I'll just give you a typical example that I get, and of course the right answer in this case is none of the calves will be phenotypically genetically affected because there's never going to be the homozygous coming together. And that is the correct answer, but I'll often get quite a high proportion that give me the 25% because we always use that example in extension talks of crossing a carrier and a carrier. And so I think we sometimes forget to go through basic Mendelian genetics when we're talking about DNA technologies and make sure everybody's on the same page. And I think this is an important point because if you do have an animal that's a carrier, it's not going to have an affected calf if you're crossing it to animals that are not themselves carriers. And so there's ways to manage around the fact that you've got an affected animal. Of course, subsequent to curly calf, we then had the identification of two other genetic recessives in the Angus breed. There was NH, or neuropathic hydrocephalus, which is kind of a big-headed phenotype, and then there's this more recent one, the curly calf, the fawn calf syndrome that came about, affectionately called CA. And basically the way that the Angus breed has responded to these is, if you look at this time frame, this is when the Angus Association recognized the genetic defect as being a defect. This is when the commercial test became available. Then this is the number of carriers that were recorded. This was current as of March 2011, and so you can see there's a fair number that have been recorded as carriers, but again I want to emphasize the fact there's a large number that have been tested that weren't carriers too, and those animals can continue to be part of the Angus population, whereas they would have been suspect if we didn't have the DNA test. Basically the way that the Angus Association is handling this is if you've got an animal with a carrier parent, you have to do testing, and basically whether you can register your heifers or bulls or not is dependent on when they're born and the results of that test. And so they have responded rapidly to these genetic defects and developed a policy to try to ensure that animals are identified so that they can be managed around. And I think really it's a major success story that DNA has been able to identify these animals and allow the management of these genetic defects. So with that background then, I'm going to move on to a little bit in terms of the project I was alluding to earlier. That's a USDA funded project looking at how to make cost-effective use of DNA information in commercial animal production. And I guess this project was really brought about as a result of, there's a lot of tests that are being sold out there for commercial producers as distinct from seed stock producers, and I was interested in looking at the economics of, well, does it pay for commercial producers to actually test their cattle using DNA tests? Does it kind of make economic sense to do that? And so I was interested in looking at how DNA-based information is best incorporated into commercial cattle ranch situations. Which of several methods of incorporation is the best? What's it feasible for commercial ranchers to do? And that's obviously one of the fun things of working in the field and trying to get some of these technologies to work. And then which provides the most or any economic benefit? And so one of the research objectives is to have a look at the traditional methods we have to look for genetic evaluation, which of course is the expected progeny differences, EPDs, that are put out by the breed associations. And then I was interested in looking at the different DNA-based genetic predictions. There was STARS originally, if you remember the very first tests that came out. There was the gene STARS. There's identity scores. Now there's Pfizer molecular breeding values. There's molecular value predictions. There's genomically enhanced EPDs. There's all these different things coming out, and I was interested in looking, well, what works the best and how can you implement them on commercial ranches? And basically the way that this study is set up is I'm working with 3 large commercial ranches up in Northern California, and then also with the commercial ranch herd that we have here on the Sierra Field Station. And I have about 125 Angus bulls that are part of this project, and we've basically used these bulls in conjunction with these cows located on the 4 different herds, and we're going to be looking at about offspring from about 2,400 cows per year to 7,500 cows over the 3-year time period. And we're going to take DNA from all of these bulls and do tests with all the different tests that are on the market, the Pfizer test, the Igenity test. We're going to use the molecular value predictions that's coming off the Meat Animal Research Centre at Clay's Centre, and just basically compare which of these gives us the best prediction of the bulls based on the actual progeny that we measured. And we're following those animals from birth all the way through carcass evaluation. So it's a fairly large project, as you can guess. And like I said, it's a beautiful place to work up there in Siskiyou County. I don't know if you've been up there much, but it really is a lovely place to work. So the 3 ranches that are involved in the operation up there is Cowley Ranch. This is Jack Cowley, for those of you that are not familiar with him. Then the Cook Ranch, the Cook Brothers, also located up there in Siskiyou County. And the Mole Richardson Ranch, also there. And all 3 of those producers are part of the Partnership for Quality, and so all of their animals go down to Harris feedlot, and then Harris is working with us to track those animals through the feedlot into the slaughterhouse so we can actually get a DNA sample from the carcasses as they're hanging to make sure we've still got the same animal we thought we had at the beginning, because once the head comes off, that's not such a simple thing. And then of course we work up here as well. And so we've spent quite a bit of time trying to get it so that we can get the DNA sample from the animals and make sure that our DNA sample is really solidly associated with an animal that's uniquely identified. And a lot of ranches, every year we have a new number 1 and a new number 2 and a new number 3 as the calving season begins. And then you've got number 1 that's down at Harris, you've got number 1 that's grazing or alongside mum, and then you've got number 1 that's gestating inside mum, and it gets quite complicated to keep track of everyone. So the way we've done that is using electronic EIDs to uniquely identify everything, and that's basically our unique identifier that we take all the way through to the slaughterhouse with us. So basically the way that the workflow goes is we take DNA on all the bulls and then we get the 50K SNP chip genotype done on them, and so that's basically looking at 50,000 little markers spread evenly throughout the genome, and that's a collaboration with Jerry Taylor and John Pollock at the Meat Animal Research Centre. We get a molecular breeding value prediction based on the Meat Animal Research Centre molecular or populations where they've trained on those populations to say, if I take a DNA sample based on what the Meat Animal Research Centre population's phenotypes say, I can predict that this animal is going to have this particular weaning weight EPD and birth weight EPD and carcass score EPD. And so we're basically, the original intent was to test it against the Meat Animal Research Centre. Since we've been doing this, we've also been running it across all of the commercial products as well, so that we can kind of see how well those all perform as well. Then in a collaboration with Dan Drake, who's a farm advisor, UC farm advisor up in Siskiyou County, we've been working on getting all of the ranch data in terms of which sires were out with which groups of cattle and which calving groups were there, and then we go up there and weigh them and EID them and DNA them. That's now a verb, I guess. So we're DNAing them for parentage determination so we can figure out which bull belongs to which calf, because these are all multi-sire breeding operations. Then those cattle go down to Harris 2 times a year for the fall and spring breeding herds that we work with up there. So we're down at Harris a lot on a Monday morning getting meat samples from those calves. And then finally, and I've got to give a lot of credit here to my PhD student Christina Weber, who gets all this stuff, puts it together, and then has a look and says, okay, how's it all working here? And we're really just kind of getting going on getting that data into the system. This is some of the equipment we've got set up where, like I said, we're using EIDs. We have a little handheld machine that can read an EID that's hooked up to a thing called CowSense so that we can keep all of the ranch records matched up with the cattle. This actually is, I think, up here. No, actually this is at Jack Cowley's ranch. So it all looks like it works really smooth. Let me tell you, it never works really smooth. We have invariably different problems. Sometimes we have a hard time even finding a hair in there that's not just totally smothered with manure and the like. But it's interesting to get it working on a commercial ranch, and I think we've actually got it to now where we're not slowing down the rancher too much when he's weaning cattle, which is usually when we're working animals and collecting DNA from them. So some of the problems we've had, the bulls never want to agree with us. We've tried all the different DNA types of collection that you can possibly think of. They all work great. Bleeding is a pain. It takes forever. Tail hair works great except when you've got really moist grass. We've done the nose swabs. They work great. I mean, I think there's a variety of ways that you can get this done. And I guess my favourite one at Cowley's one time was we were collecting tail hair and an animal ran through just no tail whatsoever. It was like a Manx cow. So then we were kind of scratching our head as to where we should go for the DNA sample on that particular animal. But the key issue is making sure we keep all of those records together. So this is kind of where we're at in our study. This is birth weight, weaning weight, in weight at the feedlot, and this is carcass trait data. Not all of our ranchers are doing birth weights. They're commercial guys, and only actually one of them does birth weights. So I mean, we don't make them do anything that they're not normally doing. But this is basically how much we've already collected in terms of numbers, and the blue is how much we have yet to do. And so it's looking much better than it did a year ago, but you can see, we've still got a large number, about half of the animals still to get weaning weights on. So they're all actually either born or in utero right now. So our anticipated finish date for this is around about 2013. So let's have a look at some of the data that we've found so far, the preliminary data. And one of the things that we're doing, of course, is using the DNA information to do parentage trace back to the sires that are in these pastures. And there's a number of positive things about multi-sire pastures, and these ranches are big enough to where they all have several bulls that are running in the pasture. But if you do have multiple sire pastures, it's known that there's higher fertility in such breeding groupings, and of course you eliminate any potential for having a sire entirely fail if you just have a single animal out there in the pasture. And you do tend to get a tighter calving season because there's just more competition out there when the cows come into heat, and so they tend to get settled more quickly. It has some benefits in terms of the number of different pastures you need, and if you are a purebred and you can sort it out using the DNA, it allows you to have don't have to have so many small pastures in order to assure that you get your sire assigned correctly. And it reduces the need to disturb newborn animals. And then what we've been doing with it is developing commercial ranch genetic evaluations. So for the cattle that are on our ranches, the bulls on our ranches, we can actually get what I call ranch EPDs, which is basically comparing the bulls within the groups on a particular ranch, and we're comparing that to the Angus Association EPDs, which we have on all these bulls. So this is just a typical example, and I've done this a lot on commercial ranches, and I see this every time, and that is that there's a lot of difference in calf output per bull. And this particular slide shows a number of different bulls. I believe this is off the Cowley Ranch. And this is this particular bull here, 5-2, had a total of 48 calves. He had 29 of them got bred in that first 30 days of the breeding season. Then he got the red lot bred in the second 30 days, and then the late breeds were the green bars at the top there. And we often see this huge discrepancy in the number of calves per bull. For sure, if you've got young bulls out with old bulls, you'll see that the young bulls are not getting their fair share bred. That for sure we can see, but other than that, it's not all that predictable as to what's causing this type of difference. But you can have the best genetics in the world in terms of EPDs on this guy, and he's not going to be doing much in terms of helping the genetic profile of your herd. If you break it down, this is the same data just shown broken into different breeding groups. So these were 5 different pastures that this particular ranch was managing in. And again, you see there's a dominant bull in each situation, and then some that just aren't getting much done. And so as a result of having followed Cowley's herd for so long, we were interested in comparing the cohorts of bulls that he bought in 1 year in terms of their total dollar return to the ranch. Okay, so we had a look at 2 different groups of animals. These were bulls that were born in 2004, and these were bulls that were born in 2005. So he bought a cohort of, 6 bulls in 2004, and this cohort of of animals in 2005. This is the average price that he received per steer. So because he's part of the Partnership for Quality, he gets a little bit of a bonus for marbling and quality grade. But this is the actual dollar return based on the weight of the animal going into the feed yard, which is where his sale point is. And you can see there's not huge differences here. A little bit, bounces around a little bit between those bulls. But the next one is what I really want to draw home. We followed those bulls for 5 breeding seasons. And this is total income that those animals returned to the ranch as a result of all of the calves that they produced over 5 breeding seasons. And I'm only looking here at steer progeny because we only followed steers through the feedlot. So you can basically in your brain double this because there's probably as many heifers that aren't as part of this equation. But the difference here is anywhere from $4,000 to close to $60,000 that those bulls differed in terms of what they brought in. This guy had 77 steers, presumably 77 heifers, over the 5 breeding seasons, whereas this guy had a total of 7 steers over the 5 breeding seasons. Really interesting data. Interesting to look at retrospectively. I can't predict which one's going to be a 4-7 and which one's going to be a 4-5 or a 5-6. But there's really, it really makes you realize also that there's no way that you're getting your genetic value out of the EP. If you paid a lot for the EPDs on this guy, he's not doing much to help your particular herd as a result of not getting a calf on the ground. So that's kind of some of our parentage data, so I'm going to say that genetic defects and parentage is working well. We are able to nail the parentage very well with the current tests that are out there, and if you're interested in doing that, I think that application works very well at the moment. The area that's maybe not working quite so well at the moment is using DNA tests for selection. And there's a couple of reasons for that. There's basically 2 tests that are out there on the market at the moment. There's the identity profile for Angus, which is basically, it's a 384 SNP test, so 384 markers spread throughout the genome of the animal. That's able to predict 21 different traits. And there's some of them are now getting incorporated into EBDs, and I'll talk about that in a minute. But this is for Angus. Everybody else, out of luck, right? That's why it's not working well at the moment, in my opinion. I mean, beef cattle breeding is about multiple breeds. It's not just about one breed. And so we've got this product for Angus. We also have this product. This is a 50,000 SNP chip. So it's got 50,000 markers spread throughout the genome that's being sold by Pfizer. And it's able to do this suite of traits. Again, it's targeted to Angus. And so the 50,000 SNP chips are out there, but what are producers getting faced with now? Well, this is just, I just picked this particular animal, Garb, predestined. Here's his Angus Association EPDs. Here's his gene star scores, so he's 2 stars for this and 3 stars for this. Here's his EPD percentile range. There's his breed average current sire breakdown. Here's the multi-breed identity profile scores on this bull. And down here at the bottom, we've got the MVPs from Pfizer Animal Genetics for this bull. So what do you reckon, guys? This is a good bull? What are you going to use? You know, I just think that's not helping the situation. That's just giving you 5 different sources of information and no real clear indication as to what's the best one to use. And this next slide blew me away. This just came out last week. So, do you consider genomics, DNA maker data, this is a beef magazine survey, do you consider it when buying herd bulls? 16% of people said it's the only thing they consider. That blew me away. How could you be making a decision on that? You know, it's just, and we've got, you know, the breed associations have been working for years getting phenotypes and developing EPDs, and we know EPDs work, and yet we've got these guys only considering DNA information. These guys, I guess I just write that off to really bad extension, are not using genetic information at all in their selection decisions, and so be it. But I guess the good news is the majority realize EPDs are still important, and and although they may look at the DNA information, they're still putting the emphasis on EPDs, and I really think that's where we need to push, and I still think that's what you want to be basing your selection decisions on. And the Beef Improvement Federation is very firm in that the way for DNA information to be used in beef cattle breeding is to incorporate the power that it brings to increase the accuracy of EPDs within the framework of breed association EPDs. And so basically to increase accuracy, not to be a separate piece of information that's used as a sale piece in the sale catalogue. And so that's the direction it's heading, and as you may know, both iGenity and Pfizer are working with the Angus Association to start to incorporate those molecular breeding values or the iGenity scores into the EPDs of the Angus Association, and that is definitely a move in the right direction. If you're interested in any of this, I encourage you to have a look at my website. I have a lot of information about how to combining EPD into DNA tests to improve genetic prediction, and I often write in Beef Magazine, which is why that was kind of interesting that there was 16% that were using DNA information only. But I want to just briefly touch on the future, because where we are now isn't where we need to be, but I think that's to be expected, okay? So I always like this slide of what, you know, the home computers might look like in 50 years' time. And these are some quotes from people who probably wish they didn't make these particular statements at the time they did. The chairman of IBM said that he thought there's a world market for about 5 computers, and Digital Equipment Corporation saw no reason why anyone would want a computer in their home. And so I don't want to be this guy, you know, in terms of my response to DNA here. And that is, you know, I don't know how this is going to be commercialized in the long run. I'm just talking about what's out there right now, okay? And I love this email. I get a lot of interesting emails, and this was an honest email that I got from a producer, and the spelling error was in the email, so I just left it there. But he was asking, why wouldn't I want to do a DNA test that costs $40 per head on all my bulls? I realise they're not purebreds, but isn't a cow when it comes to DNA testing? And that's fair enough. You know, that's a question I get a lot. No, they're not. They're not. A cow is not a cow when it comes to DNA testing. And the reason is we have this delightful diversity of breeds, and that mixes up the DNA. And so a marker that works in one breed isn't going to work in another breed necessarily. And there's really a simple reason for that, and I'll try to walk you through it. This is the 50,000 SNP chip we've got out there at the moment, right? And let's just say this is breed 1, and here's the gene that's doing whatever it is that we want it to do. It's got higher weaning weight or whatever. And in this breed, the A SNP here is associated with that gene. So if you select for an AA animal, you'll have a higher weaning weight, say. But here's a different breed where we've had some crossovers going on, so that now that good gene is associated with a T. So let's just say that's, I don't know, Hereford. So in a Hereford, you want a TT to get the good gene, whereas in the Angus, you might want an AA. Okay? And the reason for that is that 50,000 little markers spread throughout the genome, we're too far away from the causative mutation to actually be tracking it very well. So guess what we've done? You knew we were going to do it. We made a 700,000 SNP chip now. So all the research scientists at the moment are going crazy because they're all working with this 700,000 SNP chip. And the reason we think that's going to be better, although we said this would work too, didn't we? Well, we lied, is that we're going to have more frequent markers throughout the genome. And let's just say we've still got our gene here. Now we're going to have, between these two breeds, this section in common, such that if you have C in Angus, you want to select for the C here, you also want to select for the C in Hereford. And now let's open it up to many, many breeds, and the hope is, and this is where we think this will work, is let's just say the A is the actual causative mutation that's giving you the good phenotype. If you have a really dense chip like this, all of these different breeds are going to have that particular marker, and so that test is going to work across the different breeds. And that's what the hope is. We're not there yet. And so I really want you to take home the message that if those tests have not been trained in your breed, they will not work in your breed. Okay? And if you're being asked to buy them, ask the person selling them if they've been tested on your breed, because they would not be predicted to work based on the science. Okay? And so with that, I'll just say one last thing, this is an interesting situation we have. The way to get the most value back from DNA testing is for information to go back from the processor to the breeder, and from the feedlot to the breeder, so that you start to get some more information flow to make the cost of testing work for the whole chain. And that's going to require some form of data sharing in order, I think, for DNA testing to pencil. If you look at the value that's derived at the cow-calf sector, it's not enough to justify doing DNA testing in the cow-calf sector. If you look at feedlot sorting, it's not enough. If you look at what the processor gets back, it's not enough. It's only when you combine it all together that you can kind of get enough total value to where it actually pencils to do a test that costs, $50 or $100. There's no way that a cow-calf guy can make that pencil at that price. It just doesn't work. So in summary then, the current situation with DNA tests for selection, the bad news is they're breed specific. At the moment they're only in Angus. The data reporting is very varied and it's hard to interpret. And at the moment there's no independent tests to test how well that test actually works. There's no validation, if you will, going on of those tests. So you've just got the companies with their data on it. The good news is there's larger panels coming that are predicted to be more likely to work across breeds. And so the future is bright in that respect. And the other good news is we're starting to incorporate this information into traditional EPDs. And I think that's the best way to present it to producers, because 3 or 4 different pieces of information is not very helpful. So with that, I'd like to stop and thank you for your attention and answer any questions.
When you're trying to, a couple of questions, you're talking about incorporating the testing that hasn't been proven into an EPD, which is an estimate. And I think your one chart of showing the different tests says it all. That there's so much information that is not the same information, putting into other information that's an estimate. And do EPDs work? I'm not sure that they do, especially on the young animals. Now, when you get up to 90% accuracy rate, that's fine. But when you're at 10% or 20%, and you're estimated on an estimate on 5 tests that may be doing this, that aren't doing, that we don't know what they're doing. To me, I think people are getting so tied up into the estimates that they're not looking at the individual animals as they should. And I think your chart of showing the bulls breeding, to me, if you can find that gene of that bull that's breeding 47 to the one that is 7, that's probably more important to any cow-calf man than all the rest of those tests combined.
Well, I'll answer the last bit first, and that is you're assuming that that is genetic, and there's a lot of environmental influence there. If I took that best bull out of there, there'd be another one that would rise and get all those animals bred. So I don't think it's going to be as simple as a gene that's, the stud gene, if you will. It may be partly genetic, but I think there's going to be a lot of environmental influence. The advantage or the promise of DNA testing is that you will be able to increase the accuracy of young animals. At the current time, all animals from the same parents have the same genetic prediction, right? And then as they get their own weaning weight record, then you know a little bit more about them. Just like your own kids, they all have the same EPDs coming out, but then they differentiate into different beings, right? And that's the same with calves. And so the more information you start to get, the higher the accuracy of that breeding value is going to become. DNA information is meant to be able to sample the DNA that animal obtained and give you a more accurate estimate. And it depends how accurate that test is as to the DNA test, as to how much it will improve the BIF accuracy of the EPDs. If it's only explaining maybe 10% of the genetic variation in marbling, for example, it won't increase the accuracy of the EPDs all that much. if it's explaining 50%, then it actually will increase it quite a bit to you know, as much as having 9 or 10 offspring. And of course, progeny testing is really where you get high accuracy, but it takes a long time to get there. So that's always been the promise of DNA testing. What's been problematic is that we haven't had very good estimates on what proportion of genetic variation the different tests explained. We're starting to get some of that data now, and this is data that's coming out of the companies on how much genetic variation is explained by the different tests. Angus profile for carcass weight, their DNA test is associated with 29% of the genetic variation, 25% of the back fat thickness. So it increases the accuracy of those particular estimates.
Okay, I think we're going to have to go.
Yes?
And there's a lot of work done on the bull.
Yes?
I would say a lot of the feedstock guys, our biggest problem is what effort our customers keep to be their potential cows for the years to come. And so when a producer of the commercial industry and even a feedstock guy, when you got your 10 heifers there and you're deciding which ones are going to market and which ones are going to stay, is there a way to correlate that work into that? Or I think those guys need help with what efforts they ought to be keeping to be their future cows, because if their bulls are bringing in different times and they're on the same trade of bringing in a bull and they bring in the same thing or not the right thing, they're creating their own problem by the 10 heifers they kept, because the 2-year-old cows and the 5-year-old cows, they're not looking at the same thing. And if they're not going uphill on what their base cows is in the same direction as what the guys raising the bulls is, this angle goes wrong. And that's what I'd like to know how, and even our feedstock guys, what heifers should we actually be keeping? And is there a way to put that together?
Big question. Which heifers you should keep?
The brief answer is, in the proceedings, I did the calculation on what the value is for heifers. Because heifers don't contribute very much to the future of your herd relative to a bull, the value from DNA testing heifers is a lot less. And especially on a commercial situation, right? Because they're only going to produce 4 or 5 animals.
Well, they create milk and calving trouble, then that whole base is, they're 50% of the whole thing, because it's the whole group of cows that bring.
Right, I understand that, but if you're going to test all your heifers and select maybe the top half, well, okay, the top half maybe, depending on what your replacement rate is, the value that you derive from that, if you look in the proceedings, is actually not that high for a commercial guy. And so just because we don't have very good estimates on anything to do with maternal traits here, so heifer pregnancy, not available. Stayability, not available. We don't know how associated the tests are with traits that are of importance for heifer selection. Let me talk more at break or something, because I don't want to hold up the program. Okay.